gives me immense pleasure to introduce the chairperson of the session, Dr. Lakshmi T. Rao. She currently serves as a head of the Department of Neurophysiology at NIMHANS. She spearheads cutting-edge research on brain function, behavior, electrophysiology, stroke rehabilitation, and autism. Her ongoing project examines early life stress-induced changes in curiosity, risk-taking behaviors, and addictive behaviors, as well as the development of rehabilitation strategies to reduce motor dysfunction following the ischemic stroke. I would like to invite you, ma'am. Please invite the speaker.
Thank you. It's my pleasure to introduce Dr. Ratna Sharma, good friend of NIMHANS, neurophysiology per se. Just to give you a brief introduction about Dr. Ratna Sharma, she's currently head of the Department of Physiology in AIIMS Delhi. To academic in career, she is known as one of the founder of the Stress and Cognitive Electroimaging Lab at AIIMS Delhi. In the last 13 to 14 years, she has led pioneering research using high-density quantitative EEG to explore cognitive functions and their neural underpinnings in both healthy individuals and patients with psychiatric and neurodegenerative disorders. With over 30 years of experience, her work spans stress, cognition, and brain biomarkers for conditions like dementia, ADHD, and autism. Her contributions have been recognized with several prestigious awards, including the AIIMS Excellence Research Award in Basic Science. I think that's a nice credit for her. The Best Research Paper in Physiology Institute in the name of B.K. Anand at Epicon, and AIIMS Excellence Research Award. In 24, she delivered a keynote address on EEG microstates as predictors of cognitive behavior at the International EEG Microstate Conference in Geneva, sharing her groundbreaking research insights into how EEG microstates can inform our understanding of cognitive process and behavior. With this brief introduction, Dr. Sharma, please welcome you, and we would love to listen to your scientific talks.
Thank you very much. It's my pleasure to be here to congratulate Dr. Bindu for her successful and prestigious career in this prestigious institute. And thanks to the organizers who are sitting somewhere here for calling me to be part of this honor that is being bestowed to Dr. Bindu Kutty. Thank you very much. So I will be talking about the work that we have been doing in the Stress and Cognitive Electroimaging Lab in normal as well as neurodegenerative and psychiatric disorders. Although I do like to call this as a continuum, and which I was kind of dissuaded from calling in some of the meetings, but in the morning there were indications to this that it is a kind of continuum. So what we have been studying in the lab is trying to understand the interplay of DMN as well as executive networks in awake individuals when they are being exposed to normal day-to-day cognition, as well as cognitive load, and then see what is the relationship between DMN activity, attention, cognition, and then what happens in the subsequent sleep. So does increased activity, it has been reported that increased network connectivity in DMN regions is a sign of better regulator of sleep. So if you sleep better, you will have a better DMN connectivity is what is reported in the literature. So we went out on to study, before we go there, basically we were studying cognition, its load in normal and neurodegenerative conditions. But here was a student who was common to both me and Dr. Malik, and we decided to study what is the effect of local sleep on cognition, and that is how I landed up with this topic. before I move on there, I will just say that initially in earlier studies, we were trying to find out what causes error in a normal, very simple task. For example, if there is a working memory task, why you would do errors in that, and we all know that we do commit errors. So what are the EEG correlates of error in error trials in a normal subject? That is what we wanted to study first. So I will start with this, and then I will move on to the local sleep and cognition. So what we did, we picked up, or we in fact made this task. This was done for MD thesis of Dr. Surya Prakash, who was my student. We prepared this task in which we gave 3 different loads, and it was a simple picture memory sort of a task. So if you keep on increasing the load, the number of errors keep on increasing. So if you have to, just a minute. So if you have to do this load, remember these many pictures, you make errors. So we wanted to study what happens to the EEG microstates during this. Now EEG microstates are states which are lasting for about 50 to 80 milliseconds, and they are known as the atoms of thought. And in the morning, in one of the lectures, it was mentioned that the minimum kind of state that you can recognize in humans is about 30 to 50 milliseconds. So this is exactly what we are measuring. So here you see that before the stimulus appears, 50 milliseconds before that, we will try to find out the microstates of the brain. And what we saw was that there was one particular map, that is map 3, whose global explained variance was higher for error trials rather than correct trials. And then we wanted to see what are the cortical sources of this particular microstate. And what we realized that the right middle occipital cortex is the one which differentiates correct versus incorrect trials. After this, this was published in Behavioral Brain Research, and then we went on to study, is there a difference in connectivity between different regions of the brain during correct versus incorrect, or high load versus low load? So what we found, that of all the EEG bands that we studied, only theta band showed altered connectivity in 13 brain regions, 13 connections. Out of these, I think this is MAC to this. Out of these, there were 2 connections, namely right cuneus and right middle temporal, which distinguished high load versus low load, and fusiform and lingual gyrus connectivity was lower in error trials compared to the correct trials. And then we went on to study are there certain clusters, are there certain areas of the brain which which increase or decrease their activity? And what we found was there were 8 clusters which showed this kind of a difference, out of which the cingulate gyrus is the one whose activity changed in the lower bands. If you can see, the activity was higher in the error trials compared to correct, and it happens to be happening during the theta band. So the change in the activity was also inversely correlated with the error rate. So it was actually kind of signifying the relationship between error and the activity in the cingulate gyrus. So from all of this, we realized that we can have markers of error trials, which were all of these that I have just talked about. And that brought us to this question, that if cognitive load leads to errors, then, and there is a difference in the activity of certain regions of the brain during this error trial, then how is the brain handling this after the trials have been committed, or after the cognitive load has ceased to be there? Fine. So I'm just trying to tell, I'll explain it again. Like since morning, if you all were listening to all the lectures, it was a cognitive load. Okay? So during this cognitive load, which areas of your brain had higher activity, and after you finish this session, how the brain is going to handle or how the buffering of these areas is going to take place. That is a concept which has been known as local sleep. What they say is it's a complex physiological phenomena which occurs within specific regions of the brain, and it occurs, two important things is local in time as well as local in space. And it improves or it increases as the sleep pressure increases due to increase in the time that you have been awake or the load has been more. So these are the determinants of studying local sleep. So what we picked up was that, okay, we will make the person do a working memory task which is known to increase or induce local sleep. And how will we determine whether we are seeing local sleep is? We will determine the slow wave activity within the brain regions, which will be, where to find it is in this delta band, that is 0.5 to 4.5 hertz. And in literature, it has also been said that slow wave activity is related to memory consolidation, that we know very well to be happening during sleep, synaptic plasticity, as well as use-dependent recovery. So with this much of background, we did pick up this project wherein and also local sleep has been shown to be induced by all of these maneuvers, or during depression as well. So what and when we searched the literature, we found that there are various ways of induction of local sleep, out of which we took up this visuomotor adaptation task, and then we went on to study activation of specific neuronal population during wakefulness, that is while you are doing the task, and also regional changes after you have done the task. So the aim was to study the neural correlates of local sleep and its association with cognition, for which, one, we studied the effect of continuous cognitive performance on the induction of sleep, followed by letting the subject sleep in the lab, and then you study the slow wave activity in comparable areas in awake versus sleep subject, and also the cortical sources of that activity. Fine? So basically, we are just looking for which areas of the brain are showing slow wave activity following a cognitive load. So this was the task. There is a target which appears on the screen, the blue one, and then you have to move the mouse with the hand in such a way that the cursor goes to the target. So this seems like a very simple thing to do, but it gets complicated as you move on. So what happens is, after a few trial runs, we make the system run in such a way that you move the cursor and the mouse, and the cursor moves at different angles. Fine? So the angles chosen were 30 degree, 45 degree, and 60 degree. So you can imagine you're moving the cursor to 60 degree, you are moving the mouse to 60 degree, and the cursor moves to 30 degrees. So you adapt to this task, and it is cognitively challenging. In fact, there were some students, the volunteers, who took much longer than most of the students, much longer to do this. So they were anyway discarded, but this is a cognitively loaded task. So this is how it goes. So this visuomotor adaptation task was done, followed by dividing the group into there were two groups, one group in which they were allowed to doze off in the lab, and another group who was not allowed to sleep, and they were expected to sit with eyes open. So we would compare, we will look for the slow wave activity during the visuomotor adaptation task. That means while you are performing, is there a local sleep? Or after you have performed, what will happen to local sleep? So these are so this is how we segmented. So this is the time when the target appears. You take some time before the movement starts, and then you reach the target. So this is the response time during which we do all sorts of EEG analysis. So what we do is we use 128 channel EEG, and for example, here what we did was we performed the EEG analysis by clustering the areas of the brain where we got task-associated activation or deactivation. So these were the 3 areas where you can see. One was inferior occipital gyrus. When we compared block 1 with block 2, that means this angle versus this angle. Similarly, block 2 versus 3 was middle frontal gyrus. So this is an executive network getting involved. And you can see here, we are also simultaneously, so this minus this will give us this particular panel. And we have different bands, whether they are increasing or decreasing, which was either stimulus related or movement related. So this is what we are going to look at. Now this is for block 1 versus 3, where we found the inferior frontal gyrus, which distinguished the 2 areas, 2 blocks. So this is the summary of what we found. So we found 3 areas which were involved in the following physiological parameters. What we conclude, I mean, I don't have time to go into the details of why we conclude this, but it is concluded that executive network exhibits, because we are talking about we are having the frontal cortices involved, so we conclude that it is the executive network its activation during sustained cortical activity. Following this, we started seeing what is the effect on the post-task time. So we started seeing the local sleep during the non-REM phase, for which this was the 2 things done. One is qualitative analysis, for which we use the multi-tapered spectral analysis, and quantitative analysis, where we detected we tried to detect the slow waves as per the peak detection algorithm. So this is what happened in these are the representative records of the various types of segments that we got in subjects who were allowed to sleep. So you can see there is no slow wave activity here. In eyes closed, you start having this, basically this is in the range of alpha, followed by you start getting higher activation in the lower frequencies and so on. So these were labeled as, the corresponding whatever, whether it is high alpha followed by low alpha and so on. So this was a so-called just spectral analysis wherein we are looking for slow wave activity. And in the group that was not allowed to sleep, that was group 2, we did not find any change in the, we did not find the slow wave activity or lower frequencies here. Then we went on to see the cortical sources of the slow wave activity. And what we found was that in the literature it says that these are the various parameters you have to look for if you were to identify slow wave in a given EEG record. So these were the parameters that were looked for, and these are the details of how the data was processed, and what we were looking was for the slow waves. So the threshold should be more than 80 microvolts, and the total from positive to negative should be more than 140 microvolts for a wave to be called a slow wave. So these waves were identified in the whole EEG record, and then we went on to study the cortical sources of those waves using S-Loretta. So what we found in the 2 groups is what I will just explain. So in the no sleep group, these were the areas which got activated. Now here, if you can see, one area which got activated was inferior frontal gyrus, which is part of the executive network, whereas the rest are mainly the DMN network. And when we see the sleep group, here we see that there are many areas which are involved in which are part of the DMN network as well as the executive network. So what we conclude from here is intensive load leads to use-dependent sleep, higher slow wave activity in certain areas of the brain, and the maximum that we found and which were also present during the task were the middle and the inferior frontal gyri, which are part of the executive network. So what we conclude is that participants who were allowed to sleep show increased slow wave activity in regions which are part of both the executive as well as DMN networks. So intensive learning leads to use-dependent sleep, and frontal activation during recovery sleep may reflect the restorative function in cognition, while deactivation in the no sleep suggests impaired attention and executive function due to sustained cognitive load. So those who were not allowed to sleep had this deficit. So local sleep may enhance cognition by selectively restoring slow wave activity within the frontoparietal as well as default mode network. So this is what we know already, that during wakefulness there is a talk going on between the frontal executive networks and the DMN areas, which gets disrupted during the deep sleep, and this is what leads to restorative function of the sleep. So when we say that deep sleep is, it kind of takes care of the homeostatic function of the sleep, this is what we are saying, that there is no talk between the two regions. But during local sleep, what we found was, there was this talk happening between the frontal regions as well as the parietal regions or the DMN network, which will help in restoring the functions of the, or restoring the changes that have happened due to cognitive behavior. So this is the work that was done by one PhD student, Dr. Nabasami, and otherwise in our lab we are working on the QEEG correlates of cognitive functions in health as well as in disease. So with that, I thank you.
Very nice talk. Any questions from the audience side? Any questions from the audience side? Yeah, that's right.
I must congratulate you in this work because it exhibits the scientific rigor, for step-by-step rigor. I was interested in the question, as you mentioned at the end, that there is a talk between the frontal and parietal. So I was wondering if that can be established only when we do the effective connectivity, like how they communicate,
Yes.
forward, backward, all these things.
Very true. Like connectivity that we have done for the first study, somehow this is what happens when PhDs leave the country. So this is something definitely we have the data, and we will do about the connectivity between these regions. Linear lag connectivity we will do. Yeah,
Otherwise
that is one
it's
thing.
very encouraging that the frontal part and the posterior part is actually talking. Thank you so much.
But
Any more questions? Yeah.
while Dr. Raju is coming downstairs, I just want to share, for me, NIMHANS meant Dr. Desi Raju. And I was very, not this way small, but I was very young when Dr. Desi Raju was here, and he used to come to AIMS and ask us questions, so I used to be very scared of Dr. Desi Raju always. Like, still I have that, when the photo comes, I feel like standing up, So I have that kind of connection. So for many years, Dr. Raju, this Dr. Raju started coming to AIMS. I had the same feeling towards him. No, I don't want to look at him. But then once we talked and I asked him, are you related to him? He said, no. Then I relaxed. I said, okay, go ahead, ask the question, Dr. Raju.
Dr. Ratna, that's a very beautiful talk and a lot of complex things to understand. So your tasks are your visuomotor, and there is a load on visual task. So when you allow these people to go to sleep, would you expect some better performance at the peripheral level also, the way the ganglion cells function and LGN?
Very natural question. In fact, this was done. So some of those slides, I think they are hidden or I deleted them. So one more, because the time allotted to me was less, so I deleted that portion. So we did see the effect of local sleep on cognitive performance in 2-NBEC task. So the subjects performed the task before, as soon as they came to the lab and everything was attached, and after, in both the groups, after sleep or after no sleep. But the results were not very clear-cut as we would expect. So that there will be an improvement in the 2-NBEC task. There were clearly two groups. One group clearly had an improvement in both the accuracy as well as the reaction time. So, but then there was, within those same 32 subjects, there were some in which the performance deteriorated. So, so from that we cannot say that this local sleep helped the individual improve the performance. And it was very clear-cut, I mean, 50-50% sort of a thing that happened. So and same thing happened in the no sleep group also.
I thought you were going to justify afternoon siesta.
We thought so. I mean, it has been known that, this kind of sleep for short time, it will, as Dr. Malik was also trying to say, it will re-establish the deficits that have happened because of either load or continuous performance. But one group of, subjects did not show improvement. We will publish this sometime soon. But that is it. That is what it was.